Let's talk about stats, baby. In this video, I will talk about the statistical tests that we use in biology, beginning by talking about how to choose your statistical test, then the essentials about how we go about carrying out each of the statistical tests. And then what I'll do is, in a separate video, I will talk about how we would do a statistical test based on real examples. This video is really about summarizing all the statistical tests, looking at how essentially, even though each of the statistical tests has to be used for a specific type of data, but the general principles of all the statistical tests are essentially the same. And if we can identify those steps, then it might make doing statistical tests much easier. Okay, so let's begin. So what are the three types of statistical tests? First, we have the t-test. Then we have the chi-squared test, and finally the Spearman rank correlation coefficient test. So essentially it's these three tests. So let's begin with when do you use each one. So t-test is used really to look for significant differences, and I would underline the difference. Okay, so you're looking whenever you've got data and you're comparing essentially the same thing in two sets of data, you're looking for a significant difference to see if there is a significant difference, or whether actually there's no significant difference and these two sets of data are the same. You are going to be doing a t-test, usually from a mean of a population. Okay, so the type of data is usually one that I like to say is kind of x of population 1 versus x of population 2. So it's the same variable, shell size of population A, shell size of population B, the height of population A versus the height of population B. Whenever you're comparing the same variable in two different populations or two different sets of data, but it's really measuring the same thing, that's when you've got t-test. Okay, so that's the first thing. The other thing is it's the same variable. Okay, so same variable, you might have a mean, or you've got a set of data from which you can determine the mean. That's another clue that you're looking at using a t-test. Okay, so that's the first thing, that's for t-test. You're looking for differences, and essentially you're looking for differences in the same category, in the same variable. Chi-squared is, so how do you know when you're supposed to use a chi-squared? What does that data look like? Well, usually you have two or more categories. So data that fits into categories, so you might have different species, you might have different places, different locations, things like that. So you've got two or more, the data is kind of fitting into categories. Next, Spearman rank, you're usually looking for a correlation, you're looking for a significant correlation between two different variables. So I hope that we're starting, even at this early stage, we can start to see that there's significant difference, no pun intended there, there's a lot of difference between the tests. So with the Spearman rank correlation test, you're looking for obviously correlation, but you're going to have two different variables. Okay, with the t-test you're looking for difference, not correlation. With the t-test you've got the same variable. With Spearman you've got two different variables. So you want to see if height is correlated with weight. You want to see if hair color is related or correlated with intelligence, just for example. Okay, So here we are looking for correlation. There will only ever be two variables, and they will be different variables. Okay, they should usually be different variables, because you want to see if there's a correlation. Usually in biology we're looking for whether there's an effect of A on B. Okay, so that's why there usually are different variables. You want to see if one thing is related to the other thing, and if so, is there a chance that the one thing is causing the other thing, okay, or causing a change in the other thing. Okay, so two variables, and they must be different. So these are the main types of differences that you might notice in the beginning, and these are the kind of things upon which you'll have to base your decision about what you're looking for. Okay, so to summarize, two things. First, what is the kind of thing that you're testing for? If it's a difference, t-test. If it's difference from a result that you had an expectation about, or you would have expected, chi-squared. If it's a correlation that you're looking for, Spearman rank. Then with the data, with t-test, typically you will have the same variable, just two sets of those. With chi-squared, you'll have categories, and it's more about numbers of things in those categories. Okay, so we're talking more about frequencies, so how many in each category, rather than sizes and heights and distances. Okay, so if it's like with categories and frequencies and how much you'd expect, that's chi-squared. If it's two different variables, and you're looking to see if there's a correlation, association, a cause-effect relationship, although correlation doesn't necessarily imply that, if it's that kind of thing, we're looking at Spearman rank. Now let's proceed to the steps of the test. Okay, and generally speaking, I've got 4 steps. Okay, so one is to make a null hypothesis. Hopefully this is not the first time you're doing this, so this should sound kind of reasonably familiar to you. Okay, just kind of trying to clarify it, trying to show you that there's a structure to this, and there's a system by which it should make your decision-making process a bit easier. Okay, so first, null hypothesis. Second, you've got to use the equations, got to use the mathematical calculations to determine a numerical value, which we'll call the test statistic. Okay, and we'll come back to each of these in turn. 3, once you've got your test statistic, so you've plugged your data into the equation, you've got a value out of that, that's going to be the thing that you're going to use to decide whether the result is significant or not significant. So that's calculating the test statistic. Once you've got the test statistic, you need to compare that to something to know if the number that you've calculated, is it telling you that the data, that there is a significant difference, that there is a significant difference from what you expected, or if there is a significant correlation. You've got to be able to compare your test statistic to something in order to make that decision. And that something is called the critical value. And the way we get to the critical value is by first calculating the degrees of freedom. This happens for every single test. Okay, we'll come back to that again. Finally, based on what your comparison shows, either your test statistic is less than the critical value, in which case you have to make a certain type of conclusion, and if your test statistic is higher than your critical value, then you have to conclude differently. Okay, so let's go through each one in turn. The first step is to make a null hypothesis. This is the kind of unbiased position that you have to go into as a scientist, so that you're not biasing how you're looking at the data. You're letting your test tell you the significance, rather than you imbuing or looking for the data significance in the data yourself. So the null hypothesis is your unbiased position before you do the investigation. So the t-test, a null hypothesis for a t-test would state something along the lines of, so you go in assuming that there is no significant difference between my population x1 and my population x2. So if it's the height I'm looking at, there is no significant difference between the height of population 1 and the height of population 2. Okay, so that's my null hypothesis there. Then, and I mean, I would just like to restate here that your null hypothesis is essentially it's the opposite in a way, in very simplistic terms. It's the opposite of your experimental hypothesis. So if you think that plants growing in the light are going to be taller than plants growing in the dark, that is your experimental hypothesis because there's some biological reasoning behind that. However, when you're doing a t-test, you have to take an unbiased position, so the null hypothesis will be there is no difference in the height between plants growing in the light and plants growing in the dark. That's the null hypothesis. Okay, so that's t-test, null hypothesis. Chi-squared null hypothesis would be something along the lines of there is no difference between what we expect and what the data eventually turns out to be. Okay, so there is no difference between the observed and the expected values for all the categories. So because there's no difference between the observed and expected, everything is how you expected it to be. And remember, your null position is that you didn't expect anything biologically interesting to be going on. Okay, so for example, if we had a little ecosystem, we had a tree, we had a hill, and we had a lake, for example, okay, or a pond, then if I looked at the types of species or the numbers of the species in each of these different kind of mini habitats, would I expect to see a difference? Scientifically, yes, I would, because species are adapted to survive in different conditions. But my null hypothesis would be that I wouldn't expect to see any difference between the number of birds living in the hill or on the tree or in the pond. That's what my null hypothesis would be. Okay, so my null hypothesis for a chi-squared is that there's no significant difference between the observed and expected values for whatever categories of things I'm looking at. Spearman rank then, Spearman rank, the null hypothesis in this case would be that there is no significant correlation between whatever x is and what y is. Okay, so remember we had our data was organized for a Spearman rank, it's x and y. Okay, two different variables side by side in pairs. We want to see if x is related to y. Do the values of x change in accordance with the values of y? So my null hypothesis would be there is no significant correlation between x and y, even though my scientific hypothesis would be as people's height increases, their mass would increase. So that's my experimental hypothesis, but my null hypothesis would be that. Okay, so that's the null hypothesis. I'm going into doing my test with an unbiased, uncorrupted kind of mind, and I'm going to let my data then convince me or tell me if there's something going on or not. So I haven't memorized the equations, so I will come back when I have put the equations for the test statistics in these boxes. Remember, this is just a quick breeze through about, you know, the principles of statistical testing. I will do a separate video where I go through how to do the calculations for each of these tests. Okay, and we are back, and I've transferred the equations to calculate each test statistic based on the data that you start with into these boxes. So once, and remember, the point of this video is not really to talk about the mathematical kind of manipulations that you'll have to do here, but it's more about going through the structure of the test, the steps that you need to go through, okay? And really to emphasize that actually the steps for all the tests are the same, and the more structure that you can see in that, the easier it will be for you to prepare to do them. Once we've got our null hypothesis, the next thing to do is to calculate the test statistic in order to get a value by which we can decide whether the data shows any significance or not. Okay, so these equations, they use the original data to see if there is a difference, to see if there is a difference from what we expected, and to see whether there is a correlation and what type it is. These are the equations, and I will discuss those in a different video. But once we've done the calculations, we get our test statistic. And the next thing that we have to do is, because that number on its own, it doesn't really mean anything. It only means something when we are able to compare that number to the critical value. And once we compare to the critical value, we can decide whether there's significance or not. Okay? So the next step is to calculate the critical value from the data that you have. So these numbers, they're kind of relative in a way. They don't have absolute significance. What that means is that the t-value that you get, it only means something when you compare it to the data that it came from. Okay? And the way we take account of the type of data that we had to begin with is by using the data to calculate our degrees of freedom. So we're going to have a table of critical values. Okay? So we're going to have a critical value table. Yeah, now I'm sure you've used these in the past, but I'm just going to explain what their significance is. You've got a table of critical values, okay? Lots of numbers in the table. Now one of these critical values is the one that's appropriate for the data that you are using. Okay? One of these values is appropriate for the data that you have, and then your t value has to be compared to this critical value to decide whether there's significance or not. But how do you decide which of these critical values is the right one for the data that you're using? So there's two things that go into deciding which of these critical values is the right one. There's two things, so we need two sets of coordinates essentially. We need to look across the table until we get to the 5% significance level, because in biology the kind of standard accepted probability of being wrong essentially is 5%. Okay, that means you've got a 95% chance of coming to the right conclusion. Down the table, you have to look until you get to your degrees of freedom. Okay, so what we're discussing here, this applies to all three tests. All right, so how do we get to our critical value? We go across looking for the 5% significance level, and we go down looking for the right degrees of freedom. Okay, and once we've got that and we've got this, then the point at which they meet, that is our critical value. Now with that said, we'll look now, because the 5% is standard for all of the tests. That's the same. It's always going to be the same. 5% for a T, 5% for a Chi, 5% for a Spearman. So the significance level doesn't vary, but what does vary between the different tests is how you get your degrees of freedom. Okay, so next we'll look at how it is that you get your degrees of freedom, and essentially it's based on the size of the data that you have. For a t-test, the degrees of freedom, remember, that's how much we are going down the table, okay, the critical value table. Remember, we're already set at 5% going across. But the way you get your degrees of freedom for a t-test is by adding the number of values that you had in variable 1, you add it to the number of items or the number of individual data points you had in your second population, minus 2. Okay, and that will give you your degrees of freedom, and once you have got that from your data, you've got your degrees of freedom, you simply read and find your critical value based on that. For a chi-squared test, again, the principle is the same. We've got to go across to 5% on the critical value table, and we've got to go down the degrees of freedom. Now for a chi-squared test, the degrees of freedom is the number of categories. So the number of categories that you had, so whether that's 2 or 3 or 4, whatever the number of categories was, minus 1. Okay, that's your degrees of freedom for a chi-squared test. So again, once we've got that degrees of freedom, we go down that much, we go across to 5, and we've got our critical value. Okay. For a Spearman rank correlation test, the degrees of freedom, again, we go across 5%, we go down the degrees of freedom, but the way we get that degrees of freedom is, it is the number of pairs of data. So remember, for a correlation test, you're looking at the correlation between x and y. Okay, variable 1 with variable 2. And to plot that scatter graph, you always need a set of coordinates, so x and y. So correlation is usually, you're always going to have pairs of data, and so the Spearman rank correlation test requires that the degrees of freedom is your number of pairs of data. So the first thing you've got to say in your conclusion is the nature of your comparison. So in this case, t was less than the critical value, you must state that. The next thing you're going to say is, because of that, then I accept the null hypothesis that there is no significant difference between x1 and x2. Between whatever the first population was and the second population, there's no significant difference between them, and however, there is a 5% chance that this conclusion is incorrect. Okay, with 5% chance of error. So you're saying with a 95% kind of confidence that that your conclusion, based on your calculations, that's what your conclusion is. Again, parallel. If, however, your test statistic is greater than the critical value that you determined, then you must state that t is greater than the critical value. Therefore, I reject the null hypothesis and conclude that there is a significant difference between x1 and x2. Again, with the chance of me coming to this conclusion incorrectly, with a 5% chance of error. Okay, so those are the steps, and similarly we make the same comparison between chi-squared and the critical value in the chi-squared critical value tables. Okay, again we go through the process. Okay, and I risk becoming repetitive here, but at least we'll get the message home. Okay, we conclude that chi-squared is less than the critical value, so we accept the null hypothesis that there's no significant difference between the observed and expected values. Again, you know, try and make it relevant to the context. Don't just repeat what you see here, but try and put it in the context of your exam question or whatever you're applying your test to. Okay, accept the null hypothesis, no significant difference between observed and expected, again with the state that there's a 5% chance or whatever. You know, it could be that you've been asked to do the test at a different significance level, so it might be 2%, it might be 1%, in which case the stringency of the test is higher, and therefore your chance of being wrong is also lower. Okay, and again, A, B, C. If the test statistic is higher than the critical value, state that. Higher than the critical value, therefore we reject the null. Yep, that there is a significant difference between what we expected and what we observed, and that this must be due to some interesting biological phenomenon. Okay, and finally, again, state the 5% chance of error. With Spearman, remember we're concluding now. I will just kind of modify this a little bit. With the Spearman, if you do, remember that you could get your critical value, and I will just add here, special box for the Spearman rank, that with the Spearman rank, your value could be -1, could range all the way from -1 to 0 to +1. Okay, and that negative sign is not shouldn't be due to some error. The negative sign indicates a negative correlation. So make sure that your calculations don't haven't introduced that by mistake. However, if one variable is increasing while the other is decreasing, that is a sign of negative correlation. Okay, so with the Spearman rank, the closer that we get to -1 and the closer that we get to +1, there's a greater chance of a stronger correlation, and you've got to refer to this when you talk about the strength of that correlation. So the closer you get to 1 or -1, you've got to refer to the strength. So if it's here, it's a positive correlation. The closer that it is to 1, it's a strong positive correlation. If you're here in this direction, it's a negative correlation, but the closer you get to -1, the stronger that negative correlation is. And this should be reflected in your results. So again, you do compare your RS value again to the critical value, okay? And the closer you are to here, the more chance that there will be that. And again, I would advise you to be wary of that negative sign with the critical value. Your critical value tables might not accommodate negative values. I'm going to say that it should be okay for you to disregard the negative sign when you're comparing to the critical value. So just for that little bit there, you can ignore that minus sign, make your comparison to the critical value, whether it's higher than that or lower than that, but then when you come back to your conclusion, you've got to kind of then start to include the negative sign, whether and what that means for the relationship, okay, between the two variables. So again, if the RS value is less than your critical value, ignoring the negative sign, if it's less than the critical value, then we accept null. We accept the null hypothesis and conclude that there is no significant correlation between the two variables. Okay? But again, because of the level of because of the significance level of our test, we have to state what chance there is that we've come to the wrong conclusion. If the test statistic is greater, ignoring the negative sign, we must state that and then conclude that we reject the null, that there is a significant correlation between x and y. Remember, put the context of your question in at this point. There is a correlation between the number of ice creams consumed in a month and the mass of the population or the subject. Okay. C. Finally, again, the chance that we are wrong about this. Okay. But again, like here, you need to add, I think, that if there is a significant correlation, is it a strong positive correlation? Is it a positive correlation? Is it a weak negative correlation, or is it a strong negative correlation? I would put that in at this point. Okay, and that, ladies and gents, is essentially statistical testing in biology. I would take special notice of each of these stages. Okay, so this is how you make your decision, basically. The type of data that you have and what you are trying to get from that data. That's how you choose the test. But once you've chosen the test, the steps are essentially the same. You have to make a null hypothesis. You have to calculate your test statistic. You have to compare your test statistic to the critical value. So you have to work out the degrees of freedom to get your critical value. You have to compare your test statistic to the critical value in order to be able to make your conclusion. But even when you make your conclusion, you're not home and dry yet. There are specific things you have to say in that conclusion. Okay, this has been an overview of statistical testing. Now we will look at some specific examples of how we do the statistical test.